Scenario 1. Demonstrate the formation and pathway of a secretory protein that needs to be released from the cell, a secretory hormone like insulin. The mRNA leaves the nucleus through the nuclear envelope. It will then go through a ribosome attached to the rough endoplasmic reticulum and code for a polypeptide chain. After the chain is created, it is threaded into the ER lumen through a pore formed by a protein complex in the endoplasmic reticulum membrane. As the protein enters the lumen, it folds into its functional shape. As the proteins go through the rough endoplasmic reticulum, carbohydrates are attached to the protein by enzymes built into the endoplasmic reticulum's membrane. The membrane keeps formed proteins separate from proteins that are produced by free ribosomes and those that will remain in the cytosol. The folded proteins depart from the endoplasmic reticulum wrapped in membranes of vesicles. These vesicles bud like bubbles from the transitional ER. The transport vesicles then bring the proteins to the Golgi apparatus. They coalesce to form a new cis-Golgi cisternae. Golgi cisternae move in a cis-to-trans direction, with the cis being the receiving side, located near the endoplasmic reticulum, and the trans being the shipping side. Vesicles form and leave the Golgi apparatus, carrying specific products to other locations and the plasma membrane. The Golgi sorts products to target different parts of the cell. Molecular identification tags, such as phosphate groups, are added to the proteins as they move through the Golgi apparatus. Transport vesicles budded from the Golgi apparatus may also have external molecules on their membrane that recognize docking sites on the surface of the plasma membrane. The plasma membrane expands by the fusion of vesicles, and the proteins are secreted from the cell. Scenario 2. Demonstrate the formation and pathway of a lysosomal enzyme that will be used within the cell, hydrolytic enzymes found in lysosomes. Similar to the formation of the secretory protein, the mRNA will leave the nucleus through the nuclear membrane and attach to a ribosome, which will produce a polypeptide chain. As the polypeptide chain enters the rough endoplasmic reticulum, it begins to form into its functional shape. As the protein travels through the endoplasmic reticulum, carbohydrates are attached onto the protein by the enzymes on the ER membrane. These proteins are then transferred to the Golgi apparatus through vesicles for further processing. As the protein moves through the Golgi apparatus, it is tagged with the mannose-6 phosphate signal, which targets the protein specifically to the lysosome. They then leave the Golgi apparatus through vesicles and are delivered to the lysosome. They fuse with the lysosomal membrane, and the vesicles deliver its contents into the lysosome. The 3D shape of lysosomal proteins protect their vulnerable bonds from an enzymatic attack, allowing them to exist inside the lysosome. Because of the acidic conditions inside the lysosome, the tag is disintegrated and the protein is released. Scenario 3. Demonstrate the formation and pathway of a protein meant for secretion. However, it is detected as being misfolded and must be destroyed in the cell. The early stages of protein synthesis remain largely the same, with the mRNA leaving the nucleus through the nuclear envelope and the ribosome forming a polypeptide chain. Again, as the protein enters the rough endoplasmic reticulum, it begins to fold into its functional shape. However, in this scenario, it misfolds. From here, there are two ways the cell deals with these misfolded proteins. The first way is proteolysis, also known as ERAD, or endoplasmic reticulum associated degradation. In this scenario, the protein undergoes ubiquitination, where it is tagged for destruction. The protein is then unfolded and chopped up by the proteasome into short polypeptide chains. It is further broken down into amino acid building blocks by other proteases to be reused in future protein synthesis. The second method is known as autophagy. Here, the misfolded protein is also tagged with ubiquitin as well as an autophagy adapter. It is then surrounded by an autophagosome, which is a vesicle with a double membrane. The autophagosome delivers the misfolded proteins to the lysosome, where they are broken down by the hydrolytic enzymes. Scenario 4. Demonstrate the formation of a glycoprotein that will be displayed on the outer membrane of the cell for identification by other cells, such as antigens presented on the outside of the cell. Like the other scenarios, messenger RNA leaves the nucleus through the nuclear envelope, and a bound ribosome produces a polypeptide chain. The chain is threaded into the ER lumen through a pore formed by a protein complex in the ER membrane. As the protein enters the ER lumen, it folds into its functional shape. Most secretory proteins are glycoproteins. Carbohydrates are attached to the proteins in the endoplasmic reticulum by enzymes built into the ER membrane. The folded proteins then depart from the ER wrapped in membranes of vesicles. The transport vesicles bring the protein to the Golgi apparatus, where they coalesce to form a new cis-Golgi cisternae. As the Golgi cisternae moves through the Golgi apparatus, the glycoproteins formed in the ER have their carbohydrates modified. The Golgi removes some sugar monomers and substitutes others, producing a large variety of carbohydrates. The Golgi then sorts the products to target different parts of the cell, and molecular identification tags are added. The transport vesicles have external molecules on their membrane that recognize docking sites on the surface of the plasma membrane. The plasma membrane expands by the fusion of vesicles, and the proteins are integrated into the membrane.